Daniel. So, hi everyone. Thanks for being here today. My name is Olivia Kosterlitz. I'm in Ben Kerr's lab, who also will be speaking today. He's just not on yet. And I'm a fifth-year PhD student in his lab at the University of Washington. So today we're going to share a project focused on measuring the evolution of plasmid dynamics. So to start, simply, genetic inheritance is a key ingredient to evolution. The standard inheritance framework is through vertical transmission, which is where offspring inherit genetic material from the parent. And here I'm showing reproducing bacterial cells. And since it's asexual, each offspring inherits the genetic material from a single parent. But bacteria also can carry elements called plasmids, which are these small extrachromosomal DNA molecules, which encode for accessory genes. These plasmids are also inherited through vertical transmission, but this process can be somewhat imperfect, and plasmids can be lost through a process called segregation, where one of the two daughter cells didn't receive the plasmid, besides, which I'm showing here with this yellow cell. So besides vertical transmission, plasmids have an additional process through horizontal gene transfer, where plasmids are shared with neighboring non-related bacteria. And here I'm illustrating the process called conjugation. And plasmids are really common in nature. And here I'm just showing a number of cartooned bacterial communities in various environments, such as fecal matter or on an animal or in a soil or in a human gut. And as I said before, these red circled plasmids encode useful accessory genes. So when environments change, the genes on the plasmid can provide the host with an advantage. For example, let us focus on just the human gut here for simplicity. So where we can imagine now this human is consuming an antibiotic, and this environmental change will kill the bacteria that don't contain a plasmid since they don't possess the antibiotic resistance gene. And thus the plasmid containing cells survive and replicate and sweep through the population. In addition, in some cases, previously plasmid-free species can avoid extinction. If the plasmid is acquired through conjugation, then that species also will survive this environmental change. However, environments continually change, so the antibiotic is removed because the course of treatment has concluded. So now what happens? Well, we can see that all of the species now contain the plasmid, but it's no longer providing an obvious benefit. So over time, segregation will occur and plasmid-free cells will arise, but these cells don't have to maintain an energetically expensive plasmid and thus may have a fitness advantage in the non-antibiotic environment and replicate quickly, which would result in plasmid-containing cells being outcompeted. So to summarize, I have shown you that segregation, conjugation, and plasmid fitness effects all have meaningful consequences for how abundant plasmids are in bacterial communities. So it's very important for us to better understand these processes, given that plasmids play a central role in the antibiotic resistance epidemic and in the spread of and maintenance of antibiotic resistance genes. And to highlight, just this October, the US Department of Health released their second 5-year National Action Plan for combating antibiotic resistance. And this builds off the first National Action Plan, which was released in 2015. And of the 5 main goals outlined, the plan prioritizes preventing infections and to slow the spread of resistance. And a key to fulfilling those goals set forward is to have a better understanding of bacterial plasmid dynamics so we can better predict how plasmids will spread, where plasmids will spread, and how fast they're spreading through these bacterial populations. And we're all living in a current pandemic right now that has relied much on being informed by models that try to make very similar predictions, just in this case, it's specific to the COVID virus. But the COVID modeling has been imperfect, but even so, it's been a really invaluable tool for making strategic public health decisions. So this is a similar goal that we have for controlling antibiotic resistance epidemic. So for COVID, the parameter values in the models are constantly being updated to best reflect the known biology to be able to make the most accurate predictions. If this were to be applied to plasmids and antibiotic resistance epidemic, we can use parameter values in the literature, which come from a good number of studies on single species populations that contain a single plasmid. And since I've just shown you bacterial communities, we can already start to think about the limitations of this type of data. So They are useful baselines to start with, but single species studies are unrealistic with regards to the epidemic since we know these pathogens exist in communities, but currently we have a limited number of experimental studies on plasmid dynamics in bacterial communities that use numerous species and numerous plasmids. But we are starting to move in this direction, which is extremely important and the most informative scenarios for natural bacterial systems. So over the past couple of years, a handful of studies have investigated different aspects of plasmid community dynamics. This includes a study from our group published earlier this year, which is the first paper to look at the interplay of ecology and evolution in a bacterial community. And we used a simplified system with 2 species and 2 plasmid. And it revealed interesting results for the emergence of multi-drug resistance. So if you're interested in these topics, please check out some of this literature. But today we're going to focus on specifically one of the parameters important for plasmid dynamics, and that is conjugation rate, which is of central importance since it is one of the main mechanisms for spreading plasmids between different species and strains of bacteria, which helps to drive the spread of antibiotic resistance plasmids and to create new problematic pathogens. And the current methods for measuring conjugation rate have some limitations to being applied to bacterial communities, since they were developed and primarily tested on single species communities, populations, and single plasmids. So today we're going to walk through how our new method may fill this methodological gap and allow us to better characterize community dynamics. For example, to accurately disentangle cross-species transfer rate from within species transfer rate. And such an investigation like that would reveal some important information to update models and make meta predictions for plasmid outcomes in our bacterial communities. So with that, today our talk is going to have 4 parts. We'll start with the theoretical framework for our new method, which Ben will talk about. And then I'm going to come back and talk about our stochastic simulations, which is testing for the accuracy, and then finally move on to how we are doing this in the lab.
Okay, I'm hopefully going to get my screen here for you guys. And I'll make sure to switch to laser pointer here. Okay, hopefully everyone can see and hear me here.
Yes.
Awesome, thank you. So thanks, Liv, that's great. So we're going to start with talking about the theoretical side of this project. As Liv was mentioning, we're interested in measuring the rate of transfer of a conjugative plasmid. And when we're doing that in the laboratory, almost all transfer rate assays track or follow a population that has cells with the plasmid and cells without the plasmid. Plasmid-bearing cells are called donors because they donate the plasmid or a copy of it to another cell. Plasmid-free cells are called recipients. They receive the plasmid, thus completing the transfer. And most transfer rate assays involve putting these donors and recipients together and then tracking that population over a period of time where the populations grow and transfer can occur. Now, if horizontal transfer actually does occur, then we have a new cell type, these purple guys here. These guys used to be recipient cells, but they've picked up the plasmid. So we're now referred to them as transconjugants. An experimenter will track the numbers of these 3 cell types and use those numbers to put together the transfer rate estimate. But behind that estimate is a population dynamical model. Specifically, something like a system of differential equations that tracks the densities of the 3 players, D for donors, R for recipients, and T for transconjugants. So for instance, this model I'm showing you here was explored by Bruce Levin and colleagues in the late 70s. And it's a pretty good description of this system when things are well mixed and at low density. Specifically, what you're assuming is all 3 cell types here are growing at the same rate, this brown per capita psi parameter, growth rate parameter. These other terms, the green terms, if you like, are capturing conjugation, which involve a product between cells that, the density of cells that possess plasmid, so that's donors, D, and transconjugants, as well as plasmid-free cells, those are the recipients, R. And the green gamma parameter is the transfer rate. And that's the parameter we're actually interested here in estimating. you can actually, under certain assumptions, solve the system of differential equations and then use that solution to put together the transfer rate, solve for gamma, basically. And usually that's done over some period of time, which I'm going to call T tilde here. That's really often the time of the assay itself, that period of growth. So for instance, in the early 90s, Bruce Levin, Simonson, and colleagues published this result here, which is if you have the number of transconjugates at the end of T tilde, as well as recipients, donors, as well as the total number of cells, N here, and if you have an independent estimate of the per capita growth rate, psi, you have all the information needed for your gamma parameter. So this would be the estimate. Okay. This model, like all models, makes assumptions, and those assumptions can of course be called into question. One assumption, of course, is that all of these players are growing at the same rate, the brown psi growth rate. As Liv mentioned, the donor and recipient could be of different species. Also, there's often costs in terms of growth to bearing a plasmid. So this assumption of identical growth rates might reasonably be called into question. However, in the modeling framework, we can quite easily accommodate differences just by adding subscripts to each of these psi parameters, as you see here. And then, of course, we have to update the model here just to incorporate that set of growth rates now. A second assumption is also one of identicality, where you're assuming that the rate of conjugation from a donor, the red cell, to a recipient, the blue cell, is the same as from a transconjugate to a recipient. That is to say that this recipient here gets transformed into this transconjugate at the same rate as this recipient here does, that green gamma rate. But of course, we can come up with the same solution, just add subscripts here to give unique transfer rates, and then we update our differential equations as follows. So this more realistic model that I'm showing you here is going to be the model we're going to be focusing on for this talk, and even incorporating that greater realism, this model actually still is predicting a deterministic rise in the number of transconjugates given by this differential equation. However, the production of a transconjugate is itself a stochastic event, and this is especially apparent when you're dealing with low numbers of transconjugates, and that's what most assays deal with, right? So that's going to be something we're facing, and you'll see this in some of Liv's slides on her stochastic simulations. Exactly what I'm talking about here. So you're going to get variance in cell numbers, especially these low density transconjugate populations. Is such variance in cell numbers and thus variance in the estimates that depend on those cell numbers just a nuisance that we've got to put up with, or might it actually be a window, embracing that stochasticity, might that be a window to actually coming up with an estimate for transfer, these gamma parameters, in this more realistic model framework? And turns out the latter is the case, but to appreciate that, we're going to take a little historical detour back to 1943 and the famous Luria-Delbrück experiment. So this is 10 years before the double helical structure of DNA was published, that a microbiologist, Salvador Luria, and a biophysicist, Max Delbrück, published a paper with an extremely clever experiment that has now become deeply influential. And in a nutshell, this experiment revealed the nature of genetic mutation in bacteria. They entertained two models in this paper: a spontaneous mutation model where mutations occur in the absence of selective conditions favoring mutants, and an induced mutation model. That's where mutations occurred in response to selective conditions favoring the mutants. Both of their models, as well as their experiment, hinged on the recognition of mutation as a stochastic process. That's sort of fundamental to what they did. To understand that, let's think of a growing bacterial population, much like Liv outlined it, as a sort of series of doublings, an inverted tree that you're seeing here. Focusing on the spontaneous mutation model, mutations can occur at any division here. So any one of these nodes in the tree could become a mutant, which I'm just representing as purple. And I'm going to sort of have this halo around the cell if that cell itself resulted from a mutational event. That's just to distinguish it from cells that have mutations because they inherited them. So the descendants of that cell would have mutations because they inherited it from their parent. Maybe this other cell over here also resulted from a mutational event as well. Now, if we were in the laboratory and we wanted to gauge how many mutant cells there were after this population growth period that is at the bottom of this tree here, we would just simply plate that population out onto an agar dish with a selective agent, maybe an antibiotic selecting for drug-resistant mutants. And each one of those cells would then produce a colony here seen in a side view of that agar dish. However, this process of mutation is taken in this model to be stochastic, which means we could have had different nodes being the mutants, right? These two nodes could have been the mutants, right? These giving two mutant cells, giving two mutant colonies. Or alternatively, we could have had these three mutational events with inheritance that would produce 4 mutant cells, which would give rise to 4 mutant colonies. Or alternatively, let's say maybe these 3 mutational events at these nodes, which would, with inheritance, produce 5 mutant cells and thus 5 mutant colonies, and so on. I hope you get the idea that there could be variance between different plays of this population. Now that stochasticity, of course, also applies to the induction mutation model, but in a different way. The idea here is that the mutations occur probabilistically when selective conditions are first experienced. So no mutation during the growth of the population in the tree, so to speak. But when these cells hit the agar dish, some probabilistically will become mutants. Maybe these 3, or if we replate it, maybe these 2, or maybe these 4, etc. In this case, the numbers of mutants would conform to a binomial distribution, which if there's a lot of cells and the probability of mutation is very small, that would be well approximated by a Poisson distribution. The spontaneous model also has its own distribution of mutants that's different, importantly, from the induction model. And that means that if you looked over multiple replicate populations in the laboratory and looked at the number of mutants that they produced, you could use statistical signatures to adjudicate between these two models. For instance, the ratio of the variance to the mean. For the induction model, that's predicted to be unity, or 1, a value of 1, and that's a property of the Poisson distribution. For the case of the spontaneous model, that's predicted actually to exceed 1. And the reason, by the way, that occurs is because rarely you can get a mutation that's high up in the tree. And with inheritance, that leads to a lot of mutants, right? So you get a so-called jackpot population. Okay. Incidentally, the Luria and Delbrück found support for the spontaneous model. And if anyone's interested in a very nice review of both this experiment as well as some of the directed mutation controversy that followed it, Rich Linsky and John Mittler published a very fine review as well as intriguing piece in Science in 1993, I think. So I recommend folks check that out if you can. So why have I spent time with this detour to the Luria-Delbrück experiment? Well, it's an ingenious experiment, so everybody should know about it, but also because I want to emphasize that mutation is just one stochastic process of transformation. Here I just rotated our tree 90 degrees. The transformation is from a wild type to a mutant cell. But conjugation is another stochastic process of transformation. And to appreciate that, we have to look at a slightly more complicated picture of 2 entwined trees. If you can, just focus on the tree that has black edges first. And with an analogy to the first mutational case, what the transformation is here is from a recipient to a transconjugate. But of course, plasmids don't just appear out of nowhere, they get donated by other cells. So that means we have to track another population of cells, these donors that are actually giving the plasmid or donating a copy of the plasmid. To understand some of the similarities and differences between these two stochastic processes of transformation, let's think first about the rate at which transformants get produced. The transformants here are either mutants in the first case or transconjugates in the second case. Turns out that though the rate of production itself is exponentially increasing, described by this product mu times nt. So mu is the mutation rate and n is the number of wild type cells at time t. So over time, that rate of production goes up. And that's similar in the case of the conjugation stochastic process. That rate of production at the population level of transformants, in this case transconjugates, goes up here as this product. So gamma, the transfer rate times donors times recipient at respective time t. However, if we look at the transformant production per cell, per wild type cell, that is constant over time, right? So that's not going up, the rate at which any wild type cell will become a mutant. However, if we look at the same per capita transformant production rate, and this is per recipient cell here, that is not constant actually. That actually goes up exponentially. It goes up with this gamma times D, and this is because the donor pool is increasing, right? So if we wanted to think of conjugation as like a mutation process, it would have to be a mutation process where the mutation rate itself was accelerating over time. That's sort of how we have to envision it. So how do we actually, by the way, that means there's going to be some differences in our derivation of gamma that differ from how Luria and Delbrück derived the mu parameter. But there also are some deep similarities, and I'll point out some of those similarities as we go. How is it that we actually estimate this gamma parameter? Well, to give you the sort of the spirit of the idea, I was going to refer to a scene from Tim Burton's Sleepy Hollow adaptation. And in this scene, the police constable, Ichabod Crane here, played by Johnny Depp, is walking through the forest with his young apprentice, Masbeth. And Masbeth notes that like he doesn't hear anything. There's no birds, there's no crickets, there's just no wildlife, no sound at all. And that leads both of them to realize they're in sort of a supernatural presence. And that leads them to the Crone Witch, which gives them critical information on the Headless Horseman and moves the whole plot of the movie along. For our purposes here, it's the idea that the absence of something, in this case sound, gives critical information to our protagonists in this case. The same principle, I'll call this Masbeth principle, applies in the case of estimating conjugation rate. It's the absence of something. Ironically, it's the absence of transconjugates themselves that gives us critical information on the rate of conjugation. So let's apply Masbeth's principle to derive our estimate. And so there'll be a lot of math on the slide. Apologies if people tune out. It's okay. I think you'll catch the main ideas. But for those who are interested, I thought I would go through the derivation. So the dynamic variables of our system are D, R, and T, the donor, recipient, and transconjugate densities. We are focusing at some time T, here lowercase t, where we're looking at each of the population sizes of these three players. The way that they change over time is going to be sensitive to the settings of these parameters, the growth rates, the psi parameters, and the transfer rates, which are the gamma parameters. And what we're going to do is focus on a very, very small interval of time, delta t. So that means we're going to be going from this lowercase t to just a little interval t plus delta t. Now, what we're trying to compute here is the probability that the number of transconjugates over that interval increases by 1. That's what's on the left side of the equation that I'm showing you here. But graphically, we're going to have a certain number of donors, recipients, and transconjugates at the end of this little time interval. Because it's so small, however, these numbers aren't changing very much. But what we're specifically interested in is this purple pool and what's the probability of adding one more cell to this purple pool, okay? And that's what the expression on the right here breaks down into a series of terms. Each term is sort of a different way in which that can happen probabilistically. So let's focus on the first term here, highlighted in orange. This measures the probability over this time interval that a donor and a recipient find each other, that a copy of a plasmid is transferred to the recipient, thus making it a transconjugate. The donor is returned to its donor pool, and now we've got an extra transconjugate. Now that used to be a recipient, so that comes at the cost of losing one recipient, if you like. How about the next term in this expression? This is also a conjugation term. This is the probability that over this interval, a recipient and transconjugate find each other. Conjugation takes place, so you have two transconjugates, the original and a new one, that came at the expense of one recipient. And finally, this last term, this term here in the series, is simply the probability that the transconjugate cell divides. So this is the birth process. Okay, so this is the probability that we add one cell. Given that's a very, very small amount of time, the probability that we don't add a cell, that we keep the same number, is just 1 minus that, given that delta t is very small. So now we're going to invoke Masbeth's principle and focus on a case where we have no transconjugates, the absence of them, which means these purple populations here are just going to disappear. All right. So what we really want to know is sort of how would we get one of these transconjugates appearing? And there's really only one way that that happens. That's if you have a donor-recipient dyad forming, you get a conjugation event, and then you have a donor and a new transconjugate at the expense of a recipient. Now that's what we want to not happen if we want to keep no transconjugates around. So that means the probability that the number of transconjugates stays at 0 is 1 minus this term here. Mathematically, you can see that by just going up to the expression up above and plugging in wherever there's a capital T at the value 0, and you get this expression here. And by the way, anyone should feel free to interrupt me at any time if there's questions. I don't have my chat or anything above in front of me, so feel free to interrupt at any time. For notational convenience, I'm going to let probability that there are no transconjugates be denoted as P naught. And it turns out you can write P naught in a recursive way. Here, P naught over that little time interval. Oh, yeah, question. Was there a question?
I guess that's too much. I was trying to find, I'll go to headphones or I'll turn it off. You're right, it's not that interesting, except for Rob was saying we were going to do sex next. Not this year, but I'm not exactly sure what sex and bacteria means.
Sex and bacteria. Well, that's interesting. So this is, in some sense, has some of the spirit of sex as you know it in non-bacterial organisms, but the idea is actually a mixing of different sort of sources of genetic material occurs through horizontal transfer. So when you bring a plasmid into another cell, right, horizontally, you're bringing together different genetic elements, and that has an element of what we know and love about sex outside of the prokaryote domain.
Just putting on the headphones. Oh, come on.
Oops, sorry. Did that come through? Sorry, I'm not sure if you had your headphones on while I was yammering away. But maybe I'll proceed and we can come back if there's more questions. So this P naught here, right, is written as a recursion. It turns out that you can rearrange this, take the limit as delta t goes to 0, which is appropriate for a small time interval. That leads one to be able to describe the probability of no transconjugates dynamically as a differential equation. That differential equation depends on D and R at time t, the donor and recipient numbers at time t. But if you assume that those are relatively large, and Liv might come back to this, you can actually approximate, and you assume that you're at still relatively low density, you can approximate this as exponential growth, deterministic exponential growth for both of those pools. And that's what we're going to do. We're going to make that assumption. When we do that for the recipients, we're effectively ignoring the loss due to conjugation. This red X that's happening, that disappears. But that actually works just fine when you have reasonably large populations. We can solve this differential equation for an expression of P naught, the probability of 0 transconjugates, which exponentially decreases at an exponentially increasing rate. Kind of interesting. And you can see that in this expression is our gamma D. So if we just solve this expression for gamma D, we have our estimate. And that's what we do here. Now I wanted to connect this to the Luria-Delbrück estimate for mutation. And that's written slightly in a different form than they do in their paper, but this is their estimate for mutation. And you can see it's structurally very similar, right? Mutation rate is mu, that's like our gamma. The role being played by wild type growth rate, that's psi sub n, is being played now by the sum of the growth rates of donors and recipients. And the role of the initial wild type density n naught is being played by the product of donor and recipient densities. But otherwise, this is actually completely structurally isomorphic. Okay, so I'm going to just rewrite this gamma in a way where we can lose the growth rates and write everything in terms of the densities of cells at time 0 and in time t tilde, which means that we have to we're going to be looking at a growth period, again, from 0 to time t tilde here, which means that we need the initial donor number or count and the initial recipient count, as well as the final donor here and recipient count, D sub tilde and R sub tilde. And that's what's in this part of the expression. We also need an estimate, which is the hat, of the probability of having 0 transconjugates. Okay, that is that this, what I've illustrated here does not happen, that you do not produce this purple cell, this transconjugate cell. Compare that with the Simonson and Levin estimate. You can see that in the estimate is the number of transconjugates. So they're actually requiring what I've illustrated here to happen. You need to have these transconjugate cells appearing in order to calculate this estimate. Okay, so there's a kind of fundamental difference between these two estimates, which has some consequences. By the way, I'm going to call our estimate, or we call our estimate here, the LDM, which stands for low density method estimate. It also nicely stands for Luria-Delbrück method because of the inspiration here. And SIM can stand for Simonson et al. Identicality method, so that they make all the assumptions that all those parameters are identical. And that leads to this expression here. And Liv will come back and talk about the both of these metrics as well as a few others in the simulation side of things. Okay, so for our metric though, you need to actually come up with an estimate for P naught. So how do we go about that? Well, here we're going to actually again derive inspiration from the Luria-Delbrück experiment. So remember in the Luria-Delbrück experiment, they grow multiple replicate populations and ask what fraction of those populations have no mutants in them. That fraction is given in yellow here, the trees without any purple cells. That fraction is the maximum likelihood estimate for P naught in their case. So the logic for our case is exactly the same. We're going to look at a series of populations, but in this case, donors and recipients growing together. So two entwined trees that you see here. And the fraction of those populations that have no transformants, in this case transconjugates, no purple cells, again in yellow, that's going to be our maximum likelihood estimate for P naught. So in both these cases, this is the estimate for P naught with a little hat over it. What that requires in the laboratory, as Liv will show, is multiple lines, multiple populations being run out. But as also Liv will get to, that can be done in high throughput way, which is quite nice. And then in the simulation side of things, that requires multiple stochastic runs. And that is actually what we're going to turn to now. So I'm going to pass the mic back to Liv to give you guys a sense of how we approach things in simulations, as well as some of the simulation results.
All right, am I back? Okay. Thanks, Ben. So now we're going to take a detour away from math and go into some experimental and simulation results to look at the accuracy and precision of our new method. And we simulated bacterial populations containing donors, recipients, and transconjugates, and we've seen this full model before. And which I'll remind you, the derivation of our estimate and the other method mentioned, a lot of the aspects of the base model actually are removed when we make assumptions and simplify to obtain that analytical solution, such as the same growth rate, or in our case, we get rid of the fact that transconjugates and recipient transfer can occur because of the low numbers. So if we set the initial parameters, which includes growth rates, transfer rates, and cell densities, we will get this numerical solution. And I'm just showing an example here with some sets of parameters where the donors are in red and the recipients are in blue. And we can see that they're growing exponentially over time and thus increasing in cell density. And then we see that our first conjugation event between a donor and recipient occurred around 5 hours, and then transconjugates also increased over time. And if we were to rerun the simulation again with the same initial parameters, we would obtain the same result. That's because there's no random element to this simulation, which we have shown earlier is unrealistic since there's a small initial amount of those transconjugates in the population. So instead of using a deterministic simulation, which is the tool that's been applied in the past to validate previous conjugation methods, we're actually going to use stochastic simulation, specifically the Gillespie algorithm. So if we set the same parameter values as before, but run a simulation using the new algorithm, the effect is most pronounced for the transconjugate trajectory here, which we can see deviates from the mean, which we obtained from that deterministic simulation. And the stochastic simulation also only allows integers, and that's why we see these like step-like at the beginning. And the first step is showing the first transconjugate cell that was created in this population. So now we can run the simulation again and we obtain a different result. And so here the first transconjugate arose earlier in time for this population. And we can run this 100 times for each set of initial parameters that we're interested in. And then we can take the simulated populations and calculate a conjugation rate at a specific sampling time. This would be after your T tilde incubation, that's here. And here for the LDM, we pick a sampling time where we have a proportion of populations with transconjugates and a proportion of the population without. And then we can plug in these numbers into our estimate for conjugation rate and obtain our conjugation rate for this population. And we're gonna compare our estimate to other methods. And here I'm showing you the Simonson, but the sampling time is different because we need to pick an appropriate sampling time that will work for these estimates, since it's necessary, like Ben said, for the transconjugate value to be greater than 0. So it needs to be later in time. And we not only calculate the conjugation rate for the LDM, which is our method, and the Simonson one, which we showed you earlier, but we also are going to use 2 other methods for measuring conjugation rate, where TDR is the oldest, easiest to use, but it's also the most restrictive when it comes to its derivation. And the last one, the ASM, was just published this year, and it's actually an update to the Simonson method, where it actually allows there to be differing growth rates. So we'll see how that affects things. And the Simonson method has existed since the 90s, like Ben says, and it's been considered like the gold standard option. And even though I say that, I put it in quotes because it's still debated or there's confusion in the field about what to use. So the TDR, for example, is still often used in studies. The calculated conjugation rates will appear and be the true conjugation rate that we set in the simulations is indicated by this horizontal line. And then what we're estimating is the donor conjugation rate, which is on the y-axis. And then across the x-axis is a single parameter value in the model, and we'll vary that with different magnitudes, so higher and lower values. And today I'm going to show you 2 biological scenarios with our simulations. The top row, we vary growth on the x-axis. This is equivalent to adding a fitness cost, or a fitness effect of the plasmid. Either you can think of it as plasmid cost or plasmid benefit, which means that the donors grow at a different rate than the transconjugates and the recipients. And then in the bottom row, we're going to vary the transconjugate transfer rate. And this can be thought of or equivalent to having different species, where the donors are different species than the transconjugates. So in other words, the y-axis is your cross-species transfer rate, and then the x-axis would be your within species transfer rate. First, let's look at the simplest scenario where there is no plasmid fitness effect. So this is where all the growth rates are equal between all cell types. And we see that the LDM is highly accurate and precise. The TDR significantly underestimates, and I'll just remind you, this is the most restrictive modeling assumptions used. And then the Simonson and ASM, their means are on the true value indicated by that black line, but we see that there is a higher variance and thus less precision with both of these. And then when we vary the plasmid fitness effects, I was really excited to see that the LDM performs well in all the treatment scenarios. But for Simonson, we'll remember that if you differ the growth rate, you're actually violating one of their theoretical assumptions. And so we see that the mean deviates from the true estimates, but it's most notable where there's faster population dynamics occurring, and those produce the largest deviations. And for the ASM, differing growth rates was the part of the Simonson that they updated, like I mentioned. And so we see that the mean is on that gray line, but we do see a similar variance to the Simonson, so about the same precision. So we now are going to look at when we have different transfer rates for donors and transconjugates. And again, I was really excited to see that the LDM performed well in all of our treatment scenarios with high accuracy and precision. And then for the remaining methods, we know that if you differ the transfer rates, that's violating all of their theoretical assumptions for each of the 3 methods. And so we see that we see deviations from the expected value, and most notably when transconjugate transfer rate is higher than donor transfer rate. So in other words, when plasmids are transferred higher within your species than across your species. Given our simulation results, we were really encouraged to continue with our new LDM method and run tests in the laboratory. So Ben showed this before, but we start the same way as any other conjugation assay. We mix donors and recipients together and incubate for a period of time and allow growth and transfer to occur. But this is only one population in our microtiter plate. Oh, I forgot to mention to you that the transconjugates also appear there. But this is only one replicate in our microtiter plate, and so we actually have 96 of them, and this is just one of those wells. But we can take a look at another well, and we see that the replicate population, there's no transconjugates here that arose during that incubation time. And our goal is actually to be able to differentiate these two population types, ones that have transconjugates from ones that do not. So we dilute these populations with transconjugate selecting media. This would be, for example, an antibiotic that selects for the recipient species or host type, and then another antibiotic that would select for the plasmid. This would kill or arrest growth for any non-transconjugate cell type, and then we incubate, and then we'll get an array of turbid and non-turbid cultures, where the turbid cultures indicate populations where transconjugate cells arose. And then we can estimate P0, which is what we're interested in. And we can use, so now we're interested in the proportion of the populations where transconjugates don't arise. So in this case, P0 would be 0.69 because there's 66 transconjugate-free populations out of our 96. And the remaining information for the estimate, such as the initial donor and recipient densities and the final donor recipient densities, we gather by plating a few of the wells. And this is really similar to other methods, but we don't actually have to plate for transconjugates, which is the main difference. So now we apply this tool to a biological question, which is the part I'm so excited for, even though developing new tools is also cool. So before I mention our question, I'll start with a bit of background. So we tested LDM on 2 of the E. coli strains that were generated and used in this paper that I mentioned earlier. So the first strain served as the ancestor for the evolution experiment up here. And it's an E. coli host containing a conjugative plasmid, and it was evolved under plasmid selecting conditions for 400 generations. And then the second strain we tested is a clone that was isolated from the evolved lineage. And we used this evolution experiment to investigate the effect of plasmid-host coevolution on plasmid maintenance. And we test plasmid maintenance by propagating strains in non-selective media for a few days, and then track the proportion of plasmid-containing cells over time. And here we can see that they're decreasing over time. And the hypothesis is that after plasmid coevolution, you will have higher plasmid persistence in the non-selective conditions that I'm showing here. So Hannah Jort, the lead author on the paper, she ran this evolution experiment and then executed the persistence assays. And she saw that the ancestor, she saw with the ancestor that the plasmid containing cells decrease in the population over a period of 9 days. And for the evolved strain, we see that plasmid containing cells stay at a higher proportion within that same 9-day period. Thus, Hannah observed that coevolution increased plasmid persistence in non-selective conditions for our system. So you may ask, like, what changed? Why are we getting higher plasmid persistence? Well, it's typically thought that the cost of plasmid carriage is reduced after coevolution, which results in a lower competitive advantage for the plasmid-free cells. However, there is an additional hypothesis, which leads to our question: can host plasmid coevolution increase the conjugation rate? And this would be because if the conjugation rate increases, we would observe higher plasmid persistence in the population due to the increased rate of transformation of plasmid-free cells back into plasmid-containing cells over the period of these days. This is where the LDM method comes in, and we measured the conjugation rate where the plasmid containing ancestor and the evolved strain served as those donors in all of those assays we showed you. And then we had a common plasmid-free E. coli as the recipient. So using LDM, we estimated a significantly higher conjugation rate for the evolved strain compared to the ancestor. And this previous evidence that this provides evidence for us that the host plasmid coevolution in our system increased the conjugation rate. And whether this is in tandem with other changes impacting plasmid persistence, such as reduced cost, I currently don't know. But even so, the increased conjugation rate is an interesting result, because now we can use this to update our plasmid evolutionary models and factor in the importance of this trait evolving. And this may impact like our predictions for plasmid outcomes in bacterial communities. So with that, I'll just summarize that today we shared with all of you our new LDM method, and that we've successfully applied it in a bacterial system. And it revealed that plasmid host coevolution has, in our system, increased the plasmid transfer rate. And with that, I'll end with some thank yous that are much needed. I need to thank Ben. I've taken this opportunity in quarantine to get into evolutionary theory and modeling and math, and grown that skill set a bit. So I really appreciate Ben working with me closely on this project. It's been really fun. And I have to thank Hannah Jort. She was an amazing mentor for me in the lab and really got me in on plasmids, and she got me hooked. And now that's what a lot of my projects are on, and the rest of the Kerr lab for their feedback. And then I have to get out a shout out to Top Lab, which is actually another Beacon lab at the University of Idaho, where Ava has kind of made me an honorary member, which I really appreciate. And I really like their group. They're so fun. And Clint's a collaborator on this project as well. So hats off to Beacon for funding some aspects of the project, and hats off to Beacon for facilitating this collaboration, which we've done a bunch of projects with their group, and it will for sure continue after this. And thanks to my other funding sources. And thank you all for coming and listening to us.
I see Rich has his hand raised. Do you have a question?
Yeah, I do. So anyhow, that was fantastic. Thank you both for a really engaging thing. And I think Ben knows that the Luria-Delbrück experiment is my all-time favorite. And I love the way you converted it into a conjugation assay. So hats off to both of you. I guess my question is, besides sort of the mathematical assumptions that you guys have addressed so clearly, there are a couple of sort of more experimental details. And so one is sort of the instantaneous, that in the Luria-Delbrück experiment, there's sort of the assertion that the mutation instantaneously is expressed. And people have shown that that isn't necessarily true. In the case of plasmids, you'd sort of assume that instantaneously it expresses the antibiotic resistance or whatever marker you're using to select. But there's an additional assumption now that that there's also an instantaneous transfer. And I'm wondering whether you guys have thought about whether that leads to small errors, big errors. I guess it would be more relevant for the empirical things. The theory sort of says that isn't a problem.
Yeah, exactly. The theory definitely doesn't think it's a problem. So there definitely is like that time lag, like people have shown, it's like takes 10 minutes, 20 minutes, 5 minutes to get the plasmid all the way over. And we have thought about simulating that kind of into, because you can make the simulations do whatever you want and see if there's a systematic bias. I actually haven't ran those yet, but it's something interesting to look at for sure. There's also other things such as transitory de-repression. I haven't actually modeled that, like simulated that either, but that could have some different effects from just varying the transfer rate between donors and transconjugates. But it's a very interesting question and point. I don't know if Ben has things.
Yeah, I'll follow up with what Liv just said, which is for transitory de-repression, it's actually kind of interesting because that would be a case where you have potentially different conjugation rates for your transconjugates would have a different conjugation rate than your donors. And note that gamma T doesn't come up anywhere here, so it wouldn't matter. Like we would still get an estimate of gamma D, but everything Liv said, totally correct. The other, and we could build these things into models to explore whether that led to systematic bias. The other thing that Liv has noted, Hannah has noted, and others have noted is that just plating fresh donors and recipients together onto a plate that selects for transconjugates will give you colonies sometimes. So conjugation can happen on the plate, right? So if one's worried about sort of, well, is this in the midst of a transfer? And, you know, it seems as if there is actually some play temporally speaking, for these processes to play out. That's actually a problem, getting kind of spurious colony counts in some of the estimates that rely on plating. But in any case, it's a complicated situation. And I guess the first stage would be exactly what Liv said, I think, to simulate it and see what the bias was.
I think Chris Waters is next.
Thanks you both for your talk. Really interested. So way back in my PhD, I studied pheromone-inducible conjugation in Enterococcus. I'm not sure if you guys know that system, but in that case, the recipient produces a pheromone which activates conjugation in the donor cell. So conjugation rates of the donor are inherently dependent on the amount of recipient cells. So I wonder if you've thought about applying your system to that, because it seems like your new model would be really good for quantifying that system.
That's oh, go ahead, Liv.
Oh, I just have a question. So like when the recipient's around in more abundance, there's more of the pheromone, and thus you're now getting more conjugation occurring. Is that the idea?
Yeah, so actually like both the donor and recipient produce pheromone, but the donor produces an inhibitor peptide. So it keeps itself shut off. And then when the recipient's around, it doesn't make the inhibitor because that's plasmid encoded. And so it tips the balance to more pheromone and activates conjugation functions in the donor.
Interesting. Yeah, that definitely is not in the model, that specific interaction, of course. But it would be interesting because you just always get the first incidence. So you kind of get like the threshold for when it started to actually have conjugation in your system. But if it had any like amplifying effects and like kind of spiraled and sped up, you would have to do some other like assay to capture that. So it always captures the initial transfer rate that would occur. For example, that's why we can actually disentangle donor transfer rate from transconjugate transfer rate, because we never capture like when transconjugates are actually transferring the plasmid. So it would allow you to like target that system in an interesting way, different than other methods could do.
Yeah, I imagine that you could actually build it into the model where you'd have some functional dependency between your transfer rate and the recipient pool. That sounds fascinating, by the way. Just biologically, that sounds interesting. But I think you could build in. I'm not sure how what consequences that would have with regards to tractability mathematically, whether or not that would complicate things and make it difficult to come up with an analytic estimate. But in theory, that could be done, I guess.
I just wonder if you guys would get the same numbers as, because it's been studied, for 30 years. Like, would you get the same transfer rates as, what others have?
It's an interesting test that I want to do, that I'm super close to doing. So stay tuned.
I think Eric Brugger is next.
Hey guys, Eric from Idaho. Really like the talk. I was really happy that you showed some persistence data towards the end, because I had a question pertaining to your model and just these models in general, basically wondering, do any of them take into account possible loss rate? I'm especially kind of interested in the scenario where the plasmid is costly for the transconjugate, and I could imagine that it kind of could lead to some systematic underestimating of the conjugation rate that's kind of proportional to that kind of cost.
I'm so glad you asked that, because I actually have simulated segregation loss. So you're totally correct. None of the models allow you to have segregation. And so we actually can build that in the simulation, of course, and see if there's that systematic bias that you're talking about. And honestly, I was really surprised that there isn't that large of an effect. I guess one I'll say is like these rates of segregation are really high for whatever's been reported in the literature. Most people say it's 0.03 to like 0.003 to like to the -5 is, for instance. So like this stuff is more realistic. And you can see we don't get that systematic deviation. I thought it would have more of an effect, but because it's so rare and your donors and recipients are so numerous, that it doesn't really have the time to take over in that short amount of time. In a persistence assay, it's very relevant because you're transferring over days, and then you can kind of see that effect. But for the initial period, we didn't see that. And Yeah, does that answer your question?
Yeah, totally. Okay, very cool.
And then I think Chris Adami is our next question.
Yes, so my question is for Ben. You were mentioning that in your derivation of the conjugation rate, that was sort of very similar to what Luria and Delbrück did, except that your conjugation rate would be time dependent and sort of increasing in time. And then you showed a comparison between the two sort of descriptions, but it looked to me, and maybe you can just go back to that slide, as if the mutation rate estimate from Luria-Delbrück was also time dependent. And I just couldn't quickly figure out how the time dependence actually disappears there.
Yeah, I'll go back and I can share my slides real quickly here. Whoops. Can you guys see the slides there?
Yes, so.
I'll go back to the Luria to compare. Oops.
Right.
Right here.
Yeah, you see, when I was looking at that, there's obviously this time dependence down there, which is similar to. How does that cancel? As t goes to infinity, obviously the -1 doesn't matter anymore, and you just have this e to the minus psi n t coming in there.
Yeah, this term here. So as time goes on and on, this will blow up, right?
Right.
It's all positive. So this term basically approaches 0. But as you know, this term is also changing, right? So the P naught is also changing as with time in a counterbalancing way, right? So that you.
Oh, I see. See, that's the thing. I couldn't see it that would actually be counterbalancing, and if it is in fact counterbalancing, then you can say something about the P naught of t, right?
Yeah, so actually what I don't think they actually derive this in, there's a lot hidden in the Luria-Delbrück paper. They're actually using formal branching process theory in that paper, but they actually just kind of list their results, like, and so you're not seeing what's going on. For instance, I don't think they actually go through the sort of solutions for these probabilities and the differential equation descriptions, et cetera, right? That's just sort of, they just give the results at the end. But yes, you have a analytic form for P naught at time t. My guess is it actually I think this is true, that it has the same exponential form with an exponential decay, right? Exponentially increasing rate of exponential decay, right? Over time, which means that basically you're just clamping down on the probability very quickly of having no mutants. And the sort of art of doing these assays will be trying to find the window where you have some, where you have non-zero or non-1, non-unity or non-zero value for that fraction of populations, right? But yes, you can analytically get this, and it will be some expression, probably some expression exactly like this, but with the replacements that I said. So
I just wanted to thanks, that makes sense. Just share my screen very quickly, because when I took a screenshot of one of those slides, it turns out that your head was superimposed over that from Max Delbrück, so it looks like you're actually looking at Salvador Luria. But I guess I can't share my screen to show you that. I can send it to you.
Yeah, send it to you. That's great.
All right, we're running low on time, but we got a couple more people with their hands raised. We'll start with Azam Shah.
Thank you so much, Dr. Whitaker. It was a very beautiful talk, especially a lot of appreciations for initiating this kind of a model for like a horizontal gene transfer. There have been different models in the past studies, but those are based on different assumptions. But I like the spontaneous mutation attempt to basically modify the model, basically to transfer it. So I have like a 2-3 questions, but I will make it brief. Just follow-up question with Dr. Chris Waters. Like, for example, when you add a treatment in the horizontal gene transfer experiment, like a pheromone can be one example. But I am thinking about like when we give a treatment, antibiotic treatment to the patients, and then you want to apply your model in that scenario, then basically spontaneous mutation itself, I think it will be happening in the system, So the spontaneous mutation plus your transfer rate, now you have like a kind of like a add-on layer, So my question is how you are going to deal that kind of practical or clinical scenario, in your model in future. That's the first question.
Yeah, so if you're trying to look at bacterial communities that are exposed to an antibiotic, then yeah, you're asking about who's going to be resistant to who. And if there's a plasmid that's resistant, but you also can get chromosomal mutations. I took that to be the essence of your question. So the transfer rate here is really just to get you that parameter so you could make that more complex model and put in a mutation rate and put in the players of the communities that you're interested in. So you could actually take your community members, figure out how fast that transfer rate, how fast the plasmid is transferring, which is application for our model, and then use that parameter in a more complex model. That would include mutation rate.
I guess I'll just add this is sort of maybe more of a nerdy technical detail, but like when you're actually doing these estimates, right, the transconjugate selecting media, one worry would be if you had spontaneous mutations that did engender resistance in, let's say, your donor to let's say it was a chromosomal marked or resistance strain with regards to one drug, and that was the drug in your transconjugate media, you could spontaneously generate that in the donor pool, and that would be something that would also give a false signal of a transconjugant. So one would have to worry about spontaneous mutations of the kind that you're describing technically. That I think there are ways around that, but I thought I would just mention that for those who are thinking of doing these assays.
Yeah. In the paper, we deal with that by talking about pin replicating or finding your mutation rate. There's ways to correct for it anyway, but.
Thank you so much. And just like a kind of like quick question. So I think I remember like you guys just did like a study verifying this model using one plasmid type, right? It's INCP something. And being a student of plasmid biology, there is a huge variation based on the backbone gene of the plasmid. What I see in my experiments, we always struggle, like when you have the transfer rate from one strain to the other strain, it's just like a simple scenario. But when you move to the complex, like a soil microbiome, a gut microbiome, you basically totally lose the control, and it's like kind of a challenging scenario. But I think like starting with the basic and simple, this model can be used with the more modification and complex scenarios. That's a kind of a good move, I think. Yeah, thank you so much.
Thank you.
Thank you.
All right, and we'll take one last question from Rich Lenski.
Oh, so thanks. So I guess this question's for Liv. So in the 2020 paper, the Jordan et al. One, I mean, it's an interesting result that you have, and the P0 method is really nice for showing that the conjugation rate itself almost certainly must have changed. And the coevolution experiment was done in the presence of antibiotic, right?
Yeah.
So there wouldn't have been many recipients around. And so it's curious that the conjugation rate went up. So I'm wondering if you guys have any ideas. So to me, I'd say copy number, maybe something like that, that there was select because the plasmid, a higher copy would give it more antibiotic resistance or whatever you were selecting, and it might lead to a higher conjugation rate. Do you have any feedback on that?
It's such an interesting thing because we weren't expecting to see it actually, because we were like, well, there's no recipient pool around for the transfer rate to actually be selected for. So the fact that we saw it was interesting. But one other hypothesis besides copy number, I like that, is actually, but diminishing the cost of your plasmid could have some pleiotropic effect on your transfer rate, because the transfer apparatus is actually very energetically expensive. In some of our evolved lineages that Hannah had, it actually deletes them entirely. So maybe there is some mutations that diminish cost while simultaneously affected your transfer rate. I don't know exactly what cellular mechanism would do that, but that's kind of our working theory right now.
Although I do like your copy number hypothesis. Actually, that's clever.
Super.
Thanks, Rick.
All right, thank you, Liv and Ben, for such a great talk, and thank you everyone for the interesting discussion. We'll wrap it up now. We've gone over time, but it's good to see everyone.
Bye. Thank you.
Hope you all have a great weekend.
Thank you guys very much. This was great.